Namaste students, Khush aur Betiya. I JV and Dr. Vaichandra, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the diversity of microbes. Okay. So a major biological division separates the eukaryotic and the prokaryotic. The cells from animal, plants, and fungi are eukaryotes. Okay. As a true nucleus is present, whereas bacteria, blue-green algae belongs to the prokaryotes and is a primitive nucleus. Okay. So here the characteristic difference between the eubacteria and eukaryotic like fungi and protozoa or microorganism. Prokaryotic and eukaryotic, you can see the nucleus in which the nuclear structure, the prokaryotic, the circular DNA molecules not covered with protein, whereas the eukaryotes, the complex of DNA on the basis of the protein. Okay. So here the location of the nuclear structure or the dense tangles of the DNA is cytoplasm, and no nuclear membranes or the nucleoids or nuclear equivalents are present. In the eukaryote, you can find the nucleus is surrounded by the nuclear membrane. Okay, so the DNA or the nucleoid or the plasmids in the nucleus and the mitochondria is present in eukaryotes, whereas the cytoplasm, when we talk about no mitochondria or no endoplasmic reticulum or 70S ribose is present in the prokaryotes, whereas the mitochondria and the endoplasmic reticulum or the 80S ribose. So the cell wall or unusual rigid walls with murine layer or the expectation of the mycoplasm or present only or eukaryotes present only in the fungi or the glycogen or the mannose or the chitins or cellulose. Okay, so reproduction is asexual by binary transverse fission, or in most cases the sexual possibilities or asexual reproduction takes place. Okay, so here you can see the prokaryotes. The prokaryotes are like the mitochondria, or here the so when we talk about the classification, here you can find it the classification. We are talking about the identification of this separates in the groups or the interrelated area to that of the uh, so the classification or the nomenclature of the identification of the three separates but the interrelated area of the taxonomy. Okay, so here the classification can be defined as the arrangement of the organisms into the taxonomical group or the era. Okay, so here you can see the classification can be defined as the arrangement of the organisms into the taxonomical group or on the basis of the similarities or the relationship. Okay, so the nomenclature is naming of an organism by an international group. So for naming any organism, you have to follow a coding system followed by or through a proper nomenclature has been provided by a proper thing. So identification refers to the particles or the, we can see it is the isolation and distinction of a desired organism from undesirable one to verify an authenticity and specific property of a culture, to isolate the identity and casual positive agent of the disease. Okay, so it refers to as a grouping of an organism based on the genotypic or the genetic or the phenotypic observable similarities and the difference. Okay, so here when we are talking about the taxonomical classification of microbes, we have to give the phenotypic classification or genotypic classification. The phenotypic is based on the outer morphological characters, or we can see the microscopic or a morphology or a macroscopic morphology or a biotyping, okay, or a stereotyping or an anti biogram pattern. phase typing. Okay. So genotyping you can be classifying in the form of DNA hybridization, nucleic acid sequence analysis, plasmid analysis, ribotyping, and chromosomal DNA fragment analysis. Okay. So and also this numerical taxonomy is there. So you can call it as a computer taxonomy also based on the phonetic and the taxometric. Okay. So numerical classification schemes used a large number of frequency or frequently overweighting or a computer cluster. So here, according to the proposals of Rose, the world of the living thing is classified in the three domains: bacteria, archaea, and eukarya. Okay, in the system, each domain is subdivided into kingdom, bacteria, heterotrophic, and eutrophic. All human pathogens or the bacteria or photosynthetic cyanobacteria. Okay, they are not all are not pathogenic. Okay. Cyanobacteria is very useful bacteria which gives a photosynthetic activity, and this activity helps us in many of the agricultural field also. Okay. So the bacteria is classified as the reproducing asexual by the binary transfer fish. Okay. So chlamydia obligate intercellular parasite, or riccia is the obligate intercellular parasite, rod shape or to cookies or to reproduce by the binary transfer fission or the mycoplasm or the bacteria with a rigid cell wall. Okay. So the here we can see when we are giving the so the morphologically, you can find many of them as bacteria has sized into 0.4 to 2 micromole, occurring in the three basic shapes. So here, three basic shapes like cocci is circular, spherical, bacillus is rod shape, spirochetes or helicals, or cocci singly or paired, diplococcus or uh, like chain or streptococci or cluster streptococci. So thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we will resolve it in the problem solving session. Thank you.